Hi, I'm Lauren Camden from Broward College in South Florida, and I've spent the last year or so researching coral reef degradation and restoration due to climate change related matters. I focused on the rising issue of ocean acidification and its relation to calcifying marine organisms, such as coral reefs, who rely on the declining abundance of carbonate ions available to them to build and maintain their skeletons. To begin, ocean acidification is a result of increased anthropogenic or human-caused atmospheric carbon dioxide being absorbed by the world's oceans. This produces a chemical reaction that results in a lowered pH and increased acidity of ocean water. In simplified terms, when a carbon dioxide molecule is absorbed by ocean water, two positively charged ions are produced, which cause a lowered pH value of ocean water. These then react with negative bases already present in the water, such as CaCO3 or carbonate ions, which calcifying marine organisms depend on to maintain and grow their skeletons and shells. As positive hydrogen ions react with negative carbonate bases, the ocean's carbonate saturation is decreased and corals lose density, experience lowered growth, and are unable to reproduce with as much success. In this project, two papers were reviewed to explore two related objectives: investigating evidence of change in the coral reef ecosystem and identifying adaptation strategies. First, Langdon et al. Published an experiment done in the Florida Keys in 2013 to demonstrate the relationship between increased atmospheric CO2 levels and decreased calcification rates of coral. 11 samples of a combination of shallow water starlet coral and smooth star coral were epoxied to cinder blocks with sensors recording environmental data at 30-minute intervals. Random samples were incubated in tanks in situ and given a treatment regimen designed to stimulate increased ocean acidification conditions through chemical injections of sodium bicarbonate and hydrogen chloride. This treatment lowered pH value in the incubation chambers by 0.1 and 0.2 units. Calcification rates of the incubated coral samples were determined through statistical analysis. Next, adaptation techniques were examined. Lehrman et al. Published a peer-reviewed paper in 2016 detailing research and efforts committed to establishing a coral gardening program that could shift from expensive and complex engineering solutions to a more practical and efficient approach. This technique begins with a small sampling of coral fragments collected from identified donor colonies that are microfragmented in an offshore coral nursery. Microfragmentation is a restoration technique that consists of breaking coral samples into small pieces utilizing a specialized saw that stimulates tissue growth and attaching the sawed pieces to blocks. They then grow up to 40 times faster than they would naturally, and they can be supervised and maintained in a lab setting to promote survival, productivity, and vitality. Eventually, after graduating to the adolescent stage, the fragments will be outplanted onto degraded reefs. This process not only contributes to restoration efforts, but can serve in a large spectrum of academic research. Genotypically hardier corals, as well as algal symbionts, can be identified and produced in the future. The observed results of these studies are useful to direct future resource management. The Langdon et al. Study confirmed that coral samples that experienced a 0.1 and 0.2 unit drop in pH value experienced decreased calcification rates of 50 and 52% respectively. As the average pH of ocean water drops and oceans become more acidic, corals are unable to achieve net carbonate accretion, or a buildup of carbonate ions available for growth, and calcification rates drop. The Lehrman et al. Study, however, provides an opportunity to take action. Currently, there are over 150 programs in around 20 countries worldwide using coral gardening methods. The implementation of raising samples past the larval stage in controlled nursery settings and labs has helped to bypass the high mortality rates seen in early life stages and provided prospects for a greater impact on genetic diversity in the wild. All of this can be concluded simply. To combat the threat of coral extinction, coral gardening techniques are indispensable. They also contribute to filling the many data gaps present in this field. Genotypes in algal symbionts that can withstand pH and temperature change can be sought in nursery lab experiments, as well as other restoration techniques such as assisted migration, breeding, and evolution. However, these are topics that I could do an entire separate presentation on. So with that, I would like to acknowledge Dr. Pamela Fletcher, an amazing professor who has been absolutely indispensable in teaching, inspiring, and supporting me in my research and my education, as well as the research done by all the scientists before me, upon which I was able to conduct my own research. Some of their fascinating work is highlighted in this reference section.